I'm so pleased today to present Jabron Kokar, who will be giving us our very first seminar of the 21-22 Women's Health Seminar Series. And before I do that, I want to have a little land acknowledgement. I want to recognize that we all live, work, and play and participate in a community. And here in BC, we are on the unceded, ceded, and traditional territories of 203 First Nations, along with 38 Métis chartered communications, each of which possess their own unique traditions and history here on this land that we now refer to as British Columbia. We acknowledge the importance of the Truth and Reconciliation Commission of Canada's call to action, the United Nations Declaration on the Rights of Indigenous Peoples, and the BC Declaration on the Rights of Indigenous Peoples Act. In all of our work, we are committed to ensuring Indigenous women's rights to health and safety and equal opportunity to participate in a manner that recognizes and respects Indigenous cultures and traditions. Today, myself, I'm joining you from North Vancouver, because this is virtual, which is part of the unceded homelands of the Coast Salish peoples, the Musqueam, Squamish, and Tsleil-Waututh First Nations. So again, thank you so much for those of you joining. I just want to tell you very quickly about the, I went too quick, about the cluster. It's free for anyone to join. Please do join us. We now have over 300 members worldwide. We do a number of events. There's a trainee session coming up really soon, I think September 20th. That should be advertised pretty quickly. If it hasn't already, we do a number of community events. We'll be doing another one in November, talking about doing a deep dive into gender and what it means and how you can analyze and record that information. We give out a number of, we call them travel awards, but they're obviously nobody's really traveling right now. But just a little hint, you have to be a member before you can apply for them. And we've had a very good success rate, like maybe 3-digit success rate so far. So please do join and apply for those as they come up. And obviously we couldn't do this without the support of a number of individuals and communities, so University of British Columbia, but also BioTalent and some other members as well. So with that, I'm going to stop sharing my screen so Jibran can share his screen. And while he's doing that, I will introduce I'm going to call him my friend, Dr. Jabron Kokar. He's an assistant professor in the Biomedical Sciences Department at the University of Guelph. He completed his undergraduate training at Queen's University, which is where I did my undergraduate too. See, there's so many connections. Who knew about that until I get to read his bio? And then his Ph.D. in the Department of Pharmacology and Toxicology at the University of Toronto and CAMH under the supervision of Dr. Rachel Tyndale. During this time, Dr. Khokhar was awarded a CIHR Tobacco Use and Special Populations Fellowship. So you can see that he's been doing this type of work for quite a while. And then he completed a postdoctoral fellowship at the Department of Psychiatry at Dartmouth College with Alan Green. In addition to a number of awards, Dr. Khokhar has held the CIHR Post-doctoral Fellowship, as well as an NIH K99 Pathway to Independence Award from the National Institute on Alcohol Abuse and Alcoholism. His lab is at the University of Guelph, focuses on understanding the link between substance use and serious mental illness and drug use during neurodevelopment, which may impact this relationship. He has received funding. Did we lose him? He's coming again. He's received funding from both CIHR, which I'm very pleased about, as well as NSERC, which for those of you that are not in Canada, those are our sort of two main funding agencies. One, CIHR is sort of a Canadian Institute for Health Research, equivalent to NIH, and NSERC is sort of equivalent to US's NSF, as well as other sources as well. And I will say, I always like to add a fun fact, which we ask all of our speakers to give us because, long list of amazing things that they've done, but it's good to hear about them as a person. But before I add Jibran's fun fact, I'm just going to say I'm also very happy to serve with them on the Equity, Diversity, and Inclusion Committee at the Canadian Association for Neuroscience. That's where I really got to know Jibran. And I'll say, he spearheaded a number of initiatives. One to CIHR last year when they decided to cancel their spring competition and We tabled a letter, and I can tell you from I have a bit of a role at CIHR, and they really took into consideration that letter before they made their decision to reverse the cancellation of that competition. So it works. You know what? EDI committees can work. So Jibran's fun fact is that he has all of the numbers in his wallet memorized, like all of his credit cards, his license, his health card, etc. That is insane. And my hat off to you, Jabron. So take it away, Jabron.
Thank you very much. Can you all hear me and see me okay? Perfect. So thank you very much for the invite. So good to be here virtually. I was actually on the UBC campus August 20th, just for a little bit. We were visiting with my family, and so I was like, oh, just in less than a month I'll be there virtually. So yeah, great to be here, and hopefully I can visit UBC and the I don't know where the Women's Health Research Cluster is located in BC right now. So hopefully we can visit in person. And so I'll get started. Before I get started, I'd like to start off with a territorial land acknowledgement. As we gather, we're reminded that Guelph is situated on treaty land that is steeped in rich Indigenous history and home to many First Nations, Inuit, and Métis people today. I acknowledge that where I'm located today is the Mississaugas of the Credit First Nation of the Anishinaabe people, on whose traditional territory I'm currently situated, which is part of the Dish with One Spoon territory. And beyond that, I want us to all that are present here to acknowledge as scientists the role of science within society and our responsibilities as scientists towards the lands that we live in and the people of these lands. I hope that scientists in Canada can be a part of the reconciliation process, led and guided by Indigenous communities with humility, and contribute to understanding the impact of colonization on the body and mind while advocating for action and policy change. And the topic that I'm about to talk about today is schizophrenia and substance use, both of which have a disproportionate impact on Indigenous communities. And so not only do we start off with the land acknowledgement, but we also make sure that all of our research is contributing to decolonization and contributes, and we use our advocacy efforts to enact policy change, or at least push for it. And so with that, schizophrenia is a serious mental illness that affects roughly 1% of the population, but the public health impact and the burden associated with it is disproportionately larger, owing to the high morbidity and mortality in these patients, with their lifespans being anywhere between 10 to 25 years shorter compared to the general public. And the two main factors that contribute to these this mortality and morbidity are considered to be cardiovascular risk factors and lifestyle factors, both of which are contributed to significantly by the substance use in this population. The rates of substance use in this population are anywhere between 3 to 10 times higher compared to the general public. And here you can see in this table that whatever drug you choose, nicotine, cannabis, alcohol, cocaine, both the rates of use, so around 70 to 80% for any of those drugs, are at least 4 to 10 times higher than the rates of use in the general public. And even the DSM-4 criteria for abuse or dependence, these rates are at least 3 to 10 times higher. In the case of cocaine, it might even be 25 times higher compared to the general public. And this is important, and especially in the topic that we're talking about today, the series that we're talking about today, is there are sex differences in both the prevalence of schizophrenia itself and the onset of schizophrenia, with a later onset of schizophrenia being seen in women compared to men. But one factor that is sort of an equalizing factor between the two is those women that are diagnosed with schizophrenia and a co-occurring substance use disorder actually have a worsened form of schizophrenia. And that difference that's often seen in the age of onset, around 18 to 20 for men, to 20 to 23 for women, that difference is lost as well. And so there is an interaction here, but the fact remains that substance use in schizophrenia, 75% of those patients are male. And so while there might be some protective factors, when it does happen, happen, it seems to worsen the outcome for women. And so it's really important to try to understand both the mechanisms around underlying it, but also understanding where do these sex differences come from. And the part about this all is that substance use significantly worsens the course of the clinical disorder, resulting in treatment non-compliance, which is already an issue in these patients, hospitalizations, relapse, violence, and even increased suicidality. And all of those then contribute to the lower lifespan and the increased morbidity and mortality that I was talking about earlier. So the title, as you saw today, is the chicken or egg problem about substance use and schizophrenia. Which came first? On one side, you have the chicken. What is it about schizophrenia that makes these individuals more vulnerable to substance use and to continue using substances despite the negative consequences beyond the negative consequences that are seen in the general public? And, you know, oftentimes I'll ask the crowd, why do you think patients with schizophrenia use substances? And the answer, you know, it's a little harder to do on Zoom, but the answer that I've gotten 100% of the time is the self-medication hypothesis, that they are using these substances to medicate themselves. When we look at the data, that's not entirely supported. Firstly, you don't actually see a clear correlation between symptom severity and amount of substance use. In fact, patients with the most severe negative symptoms are protected against substance use. Secondly, these individuals use substances often and most of the time start using these substances prior to the onset of the first episode. And then lastly, as we were talking about in the last slide, treatment non-compliance is an issue for drugs that do work. And so here, these are drugs that don't necessarily help manage the course of the disease, so compliance with it might not make sense. On the other side of the coin, or the egg side of the equation, is what is the effect of substance use on the course of schizophrenia? While I mentioned all those associations that I mentioned on the first slide are associations, we don't know necessarily if they cause a worsening of the disorder, or they're just two distinct populations, those with substance use and those without substance use, and those are just going down two separate paths. Then, sorry, I just saw the chat pop up and I thought there was a question. And then on top of that is data, you know, longitudinal epidemiological data, that there may be an impact of exposure to drugs during critical neurodevelopmental periods like adolescence that then make somebody more vulnerable to not just the substance use, but also the development of schizophrenia. And so these are the two sides of this chicken or egg question that we're trying to solve in the lab using animal models, with the goal of then using this information to develop new treatments for use in these difficult to treat populations. So I'll start off with the chicken side of the question first. And here we proposed a unifying hypothesis now a couple of years ago, or 3 years ago, where we suggested that it's not that substance use is necessarily causing schizophrenia, or that schizophrenia itself is contributing to the substance use in some way. It's that they're probably arising from a shared genetic susceptibility that makes individuals with schizophrenia, because of the overlap between the circuitry that plays a role in reward in the brain as well as in schizophrenia, that those dysfunctions in those patients make them more vulnerable to substance use, and that's what might be driving substance use in this population. And so the first evidence of this we got was with a brain imaging study that we did back at Dartmouth, where I did my postdoctoral work. And in it, what we did was had patients with schizophrenia and a co-occurring cannabis use disorder, or healthy controls, and then they went through what's called a resting state functional connectivity study, where they were scanned, their brains were scanned for bold activation functional MRI, and we looked at how one part of the brain connected with another part of the brain. In this case, the part of the brain that we were interested in was a part of the brain reward circuit, the nucleus accumbens. And then they were scanned, and then 2 weeks later they were allowed to come back and then scanned again after either smoking a joint inside the magnet or taking a pill of THC, and then their brains were assessed again. And what we found was that the controls are up here, and all those yellow-red blobs that you see at the front is connectivity between the nucleus accumbens and all of those brain regions. Suggests essentially functional connectivity can be thought of as dancing with a partner. And if you're any good, then you would be sort of jiving along with your partner in step. And when you move right, they move right. And when actually, if you're depending on the dance, they might be moving left, but you're in sync. But if you're not any good and you're stepping all over each other's toes and not really in sync, then you're thought to be functionally uncoupled. And so that's how I think of functional connectivity in my brain. And so what we see in the controls is there's a lot of yellow and red and orange blobs there, which means that there's a lot of connectivity between this part of the brain reward circuit, the nucleus accumbens, and these frontocortical regions here. When we look at the patients, a lot of those red orange blobs are missing, suggesting that the connectivity between the nucleus accumbens and these prefrontal cortical regions in these patients are significantly reduced. Once again, I should mention that here, the patients had been using cannabis for at least 13 years. Most of these patients were male because it's extremely difficult recruiting females into these studies owing to the 3 times higher rate of substance use in the co-occurring population in males compared to females. And so the bottom just shows the difference between the controls and the patients. And this here, it is tabulated. And we're just looking at the nucleus accumbens to the medial prefrontal cortex. And so in the controls, the first scan and the second scan don't look any different. But then when the patients who have this reduced connectivity compared to the controls, when they either smoke the cannabis or take the THC pill, we see an improvement in their connectivity. And so this led us to the hypothesis that maybe this dysfunction in the brain reward circuit is what is driving the substance use. And the substance use may in fact be correcting some of this dysfunction while still making worse the course of the schizophrenia in these patients. But this is a study done in humans that had been using cannabis for at least 13 years. And then for the course of this study, we're asked to be off of their cannabis and other drugs for at least a period of 3 weeks, some up to a month. And so it could be that this difference or this change that we're seeing is an effect of withdrawal, some other effect that may have occurred as a result of their long history of substance use. And so when I got to Dartmouth, my goal was to see if we could get at some level of causality here, but also assess whether the previous exposure to drugs or any of these other factors were contributing to the development of this brain reward circuit dysfunction. And so I turned my attention to animal models, being an animal model-based neuropharmacologist, and looked at all the models that existed for schizophrenia. And the one that kept coming up over and over again is this neonatal ventral hippocampal lesion rat. And the reason it came up is not only because it's used often to model schizophrenia, but in addition to showing negative cognitive and positive like symptoms that are seen in patients, it also shows increased intake of nicotine, cocaine, methamphetamine, and alcohol. So it has face validity on that dual diagnosis or co-occurring disorder part of the equation as well. And so what we did was we took these animals, did that surgery on day 7 in these animals, and then allowed them to grow up. Since patients with schizophrenia often use substances prior to their first onset of the schizophrenia, we allowed them to use alcohol during adolescence, and then they were exposed to alcohol again in adulthood. And then the other thing we tested was the predictive validity for this animal model. So we treated it with either clozapine, which is an atypical antipsychotic that is known to reduce alcohol and substance use in patients with schizophrenia, and compared it to a typical antipsychotic haloperidol, which does not reduce substance use in patients with schizophrenia at all. And what we found was that the NVHL rats drink almost 3 times more alcohol than sham rats. So the dotted lines that you'll see here are the animals with the lesions. So the lesions in the line equal the lesions in the hippocampus, right? And then the solid line is the control. And we see that over time, these NVHL rats drink 3 times more alcohol than sham rats. But this is male rats. And so when we look at female rats, we actually see a very different pattern. And so here you can see the red line versus the black line. Both of them with the dashed lines are the NVHL rats, and then the solid lines are the sham rats. And we see that while both the NVHL rats and both the male and female NVHL rats show more drinking than their sham control counterparts, their patterns of drinking look very different, where the males are actually starting off low and they increase to the point of, being around 2.5 to 3 times higher than the sham animals, whereas the females start off high and then level out to the same place. And so this is really interesting to us, and we're following this up with, and not to give the punchline away, with brain imaging studies to try to understand what the sex difference is, and is this in fact, I have no misgivings about whether this model is exactly like patients or not. And so it is a model, all models are flawed, but whether this is in fact related to those sex differences that we observe in the patients as well. So we see that these rats achieve moderate levels of alcohol compared to sham animals. And then getting to the predictive validity part of it, we see that when treated with clozapine compared to vehicle or haloperidol, these animals, they're drinking flatline. They stop drinking, much like what's seen in the patients, in that the patients show reductions in alcohol drinking and substance use after clozapine treatment. And we see the same thing in this model as well. And it's interesting, there's always a question, what is the drug doing to these animals? Are they just now lying flat on the side of the cage? And some antipsychotics do that, so then they wouldn't obviously be drinking. But when we take away the clozapine, it takes almost as long, or 25 days or so, for their alcohol drinking to come back to where it was before the treatment started, just like it initially took them that long to ramp up their drinking. And so we're trying to understand what it is that clozapine changes. And because unfortunately, while clozapine works, it's probably it's one of the more notorious drugs on the market in terms of it having a black box warning. It has significant side effects associated with it, so it's not used all that often in patients. And so we'd love to figure out what it is about clozapine that makes it so, and then hopefully come up with a safer treatment that can achieve the same effect. So now what I've shown you so far is that this animal model has some face validity in the behaviors that it performs or look similar to what's seen in the patients. It has predictive validity in that the drugs that work in the patients also seem to have an effect in this animal model. Theoretical validity in that it's based on that neurodevelopmental model of schizophrenia. But every time I write a grant on this topic or a paper, they come back with, we can't model schizophrenia in animals, let alone you're trying to model schizophrenia and something else on top of that in animals. And so what we did was decided that we would look at the brains of these animals, that if it walks like a duck, talks like a duck, and its brain scan looks like the duck's, then maybe it's not a bad model for the duck. And so we did the exact same thing. And so I'm realizing that my Zoolander references are getting kind of dated now. My class, nobody knows who Zoolander is. So, Zoolander, yes, we did use an MRI for rats, and it works very similarly to the MRI for humans, except that it has a small bore and it's a higher field, so you can get quality images of the brain of a rat. And what we did was scan the animals prior to that separation of alcohol drinking. Before ever having that alcohol in adulthood, we performed the fMRI scans to remove the possibility that it was their extended duration of substance use that was resulting in that brain reward circuit dysfunction. And the great thing is we are able to use the exact same analysis methods that we did in the humans in these animals. And so once again, we took the nucleus accumbens as the seed. This is the front of the rat brain, back of the rat brain, and those blue blobs that we see here are reduced connectivity in the NBHL rat compared to the sham animal. And we see the exact same brain regions, the cingulate cortex, the prefrontal cortex, the orbital frontal cortex, the insular cortex, showing decreased connectivity in the NBHL animals compared to the sham rats. And so just like we had done with the human graphs at the beginning, you see this decreased connectivity between those, the nucleus accumbens and those brain regions. What was important for us is that, yes, now we've seen it in animals. Yes, it occurs before they drink alcohol at high levels. But to be able to manipulate it and causally establish that this contributes to their alcohol drinking, we needed to be able to manipulate the brain circuitry that's underlying it. And one thing that's important there is to look at what the direction of the anatomical afferents are. And those go from the prefrontal cortex to the nucleus accumbens. And so when we look at the connectivity that's going backwards now with the seed and the medial prefrontal or the prelimbic cortex, we see backward reduced connectivity with the nucleus accumbens. And so now we're modulating this connectivity using chemogenetics or designer receptors exclusively activated by designer drugs to modulate this while the animal is in the magnet and while behaving to establish whether this can contributes to the alcohol drinking in this animal. And so hopefully the next time I'm here, I'll be able to show some of that data. What's important here is to realize that this is not just a brain-wide hypoconnectivity, that this is, that the brain is just not communicating as well. This seems to be specific to the brain reward circuit, because when we look at a seed in the retrosplenial cortex, we do not see any differences between the NBHL and sham animals, even though we see really nice connectivity maps for the retrosplenial cortex to the frontal cortex or the hippocampus regions that it's known to be anatomically connected with. We don't see any differences between NBHL and sham rats. And that's a good thing, because now it gives us a specific place where we can go in and manipulate to see if that's related to the behavioral outcome. So what we've shown so far is that this animal model not only does it have those three types of validity that we were talking about earlier, but also has diagnostic validity, in that a measure that we have gotten in patients is also observed in these animals. And hopefully we can then manipulate the circuit enough to be able to modulate these and then establish whether they're causally contributing to the behavior. And so now we'll flip, and I know I'm running short on time, but we'll flip over to the egg side. And this is also a lot of where the fun, cool findings have come out of in the last 3 years in the lab that I wanted to touch upon before in this talk. And so we'll start off. This is something that we've done in the lab, and so this is a plug, and I'll show you a video of it later. But we've been working on a lot of open source tools in the lab to help make it easier, cheaper, faster to be able to do these experiments in the lab. And so one of these is an exposure chamber that allows us to expose rats to vapor from nicotine, cannabis, whatever you can vaporize, we can expose rats to it. With this, we recently did another study with the NBHL rats and looking at what those causal effects of now substance use itself is on the circuitry in these animals. And so we took NBHL rats or sham animals and implanted electrodes for local field potential recordings, and then exposed them to either two different types of cannabis. One cannabis that had balanced cannabis or almost equal amounts of THC and CBD, and then a high or THC only type of cannabis that did not have equal amounts of CBD in it. And so these are, compared to before, which was functional MRI studies, these are electrical studies of oscillations within the brain, of summed electrical activity in these brain regions. And what we see is that both the NBHL rat surgery as well as the cannabis exposure differentially modulate this brain activity within the brain. So if you look here, we see that compared to sham animals, the NBHL rat shows this gamma hypo power or decreased gamma power compared to sham animals, which in fact seems to be worsened by the THC only exposure, but ameliorated to some extent by the exposure to the CBD plus THC. So what we're trying to establish here is, by changing, if they're going to continue using cannabis anyways, maybe we can change the type of cannabis and reduce the harm that's associated with this cannabis use. And we see similar patterns with the nucleus accumbens and the hippocampus as well. Once again, that decreased gamma power in the NBHL rat at baseline without any drug exposure, and then when THC is given, it's still there, but then with the THC plus CBD, some of that is ameliorated. And it's the same thing here in the nucleus accumbens. We see that decreased gamma power significantly worsened by the THC exposure, but then almost returned back to where it is in the sham animals, in the animals that receive both THC, CBD and THC, the balanced cannabis. And so this is only in male animals, and we're currently repeating this in female animals right now. And so we see a similar effect on coherence, which is once again that measure that assesses how one activity in one brain region correlates with the activity in another brain region. And we see that while we don't see much of an effect in the gamma range, we do see some effect in the delta range, where both types of cannabis worsen the cingulate PFC delta coherence, which has previously been linked to cognition and cognitive outcomes. So it's possible that while THC and CBD might improve signals in one part of the brain, they may still be worsening them in another. And that's the sort of balance that we need to strike when figuring out what cannabis use, if any, is beneficial or harmful for patients with schizophrenia, and how to help reduce the harm associated with it. And then the next thing that we looked at is this cross-frequency coupling. And so we see in the NBHL rat compared to the sham animals that there exists this 20 millisecond delay across multiple brain regions, and both NBHL, both the THC and the CBD treatment in the NBHL rat seems to correct that delay. And so, as we were talking about, it's not a one-size-fits-all effect of cannabis. While it may worsen synchrony, it might improve cross-frequency coupling while still reducing power, or the power might be differentially impacted by the amount of CBD in that cannabis. So now we'll move on to genetics. And the reason genetics are also important is, as I started in the beginning, I was talking about that shared genetic susceptibility that gives rise to both. And so we know a little bit about the genetics of cannabis use and the genetics of schizophrenia. And so this is from a recent review that we published. We've been looking at the different targets that might be contributing to both in a sort of a transdiagnostic manner. And this gene, the CADM2, which is the cell adhesion molecule 2, it's been previously associated with drug use, metabolism, cognition, and neurotransmission. And so we've been trying to establish whether this gene contributes both to substance use, but also to schizophrenia and other serious mental illness related symptoms in patients from genome-wide association studies. So we've established, in collaboration with Abe Palmer, a knockout model of this gene and have been testing it for a variety of outcomes. And I'll just briefly touch upon one of them, and then we can talk about others if they come up in the questions. And this is using an edible-based approach. And so we've been using either pure THC or cannabis oil that we've purchased from the Ontario Cannabis Store, and we make this cookie dough in different colors and let the rats or the mice self-administer it. And so we found some pretty cool findings that especially have a really nice sex difference in them. And so I wanted to highlight that as well today. So when we look at, so in the genome-wide association studies, what we find is that the CADM2 genotypes with higher activity are more likely to be associated with increased cannabis use or increased risk of lifetime cannabis use. So here, when we knocked out the gene in And I seem to have lost my legend. In here, what we're seeing is that the black lines here are the wild types, the red are the heterozygous, and then the green are the knockouts. And what we see is that the knockout animals with the CADM2 gene show a significantly lower preference. So how much of the THC dose they consume compared to the control dose. And we see similar findings between males and females, but it appears that the females seem to, at least this is with the pure THC, this is not significant, but we're seeing a trend for possibly increased preference for the cannabis dose or the THC dose compared to the males. And when we look at the cannabis oil dose, so in the previous slide, it was the THC, pure THC itself. Here, we've taken all the other ingredients in the cannabis oil and the terpenes, et cetera, and those are a part of this dose. And we see a similar effect again with the significantly lower significant effective genotype with significantly lower preference for the cannabis dose in the knockouts and the hets compared to the wild type control. And here we see a significant effect of sex as well, where the females, especially the control animals, show a significantly higher preference for the cannabis dose compared to the male animals. And that's in fact consistent. We don't have much data looking at sex differences, but the little data that we do have suggests that while all other forms of cannabis use are significantly higher in males compared to females, when it comes to oral use of edibles, et cetera, those are significantly higher in the females compared to the males, and we're recapitulating that in our mouse model. And so we're trying to now, using fiber photometry and MRI imaging, trying to both figure out what the impacts of that CADM2 genotype are, but how it's also modulated by sex. And then the last thing we'll quickly touch upon is another drug. I wanted to give a little bit of, whatever everybody's favorite drug of choice is, I wanted to be able to cover it. And so we've covered cannabis, we've covered alcohol, and so we'll touch on nicotine and vaping a little bit as well. And so here's a video of the rats being exposed to vapor from that Juul, that apparatus that we were showing earlier. And this is connected to a Juul pod. So here we're able to use exactly what's being used on the streets. Juul, even after a ton of lawsuits, remains the most popular e-cigarette brand on the market. And so we're able to expose animals to it and then assess what the effect of developmental differences or sex differences are and how those relate to what's been observed in humans. So the first thing we have shown is that you can produce pharmacologically relevant plasma levels of nicotine from short, very short exposures to Juul vapor. So 2 minutes, 4 minutes, and 8 minutes. So, this is what would be equivalent to one bout of use in a human adolescent. And so even those short durations produce plasma nicotine and cotinine levels that are pharmacologically relevant. And then we can assess what the behavioral correlates or the age-related correlates of that use are. So the first thing we did was, do animals find Juul vapor rewarding, and would they prefer to be in a chamber that they were exposed to the Juul vapor compared to a chamber that they weren't exposed in? And so we did what's called a conditioned place preference study, looking at the effects of either nicotine vapor or vehicle vapor, which was the propylene glycol and vegetable glycerin that's usually found, the nicotine is dissolved in, plus the flavorant that's included in there. In this case, it was a mango Juul pod flavoring. And then assess them for which chamber they preferred after those exposures. And we see some really interesting data. So here on the my left, you can see the males, and on the right, you can see the females. And what we find is that both male and female, both adolescent and adult male rats show preference or significant preference for the Juul nicotine vapor, with the adolescent males showing significantly more preference compared to the adolescent males, but they still show preference nonetheless. in the females, we see that the female adolescents don't show any preference for the nicotine vapor paired side, whereas while the female adults still do, they're showing this almost like a rightward shift of the curve, where the dose that was aversive in males seems to be rewarding in females. And so we're trying to expand this further to see if there's any doses that are found to be rewarding by the adolescent females, but thus far, it doesn't seem like there are many doses that are preferred by female adolescents. And it's interesting because when you look at the qualitative data that's coming out of adolescent Juul users, it seems that the males tend to use it for more of the withdrawal relieving or the rewarding effects of it, whereas the female adolescents report using it as a stress coping mechanism. And so we're now doing some studies trying to figure out if you stressed out the females, would they now prefer the Juul vapor exposed chamber compared to the vehicle one. And then, going on to withdrawal, we see very similar findings in that both male and female, both adolescent and adult male rats show significant withdrawal. So in this case, what we did was 3 times a day, the animals were exposed to 10 minutes with the Juul vapor for 14 days, and then they were injected with mecamylamine, which is an antagonist of the nicotinic receptor that precipitates withdrawal from the nicotine. And so animals show withdrawal signs, and we see that while males show significant withdrawal from the Juul vapor, we're not seeing that effect in either the adult or the adolescent females. And so we're now using brain imaging. We're trying to figure out what are these developmental and sex-related differences that contribute to the differential responses between the sexes and between the ages. And it seems, and not just this study, another study right now that's under review, we looked at the effects of both alcohol and nicotine vapor exposure during adolescence, and it seems that the males are a lot more susceptible to the effects of that nicotine vapor compared to the females, which they seem to be protected in some ways. And so here again, looking at the locomotor effects of the Juul vapor, we see that both male adolescents and adults show increased locomotion after they've been exposed to the nicotine vapor, but this is not seen in adults and adolescents. And so not only are we looking at the brain, but we're also looking at plasma and brain levels of nicotine to see if this is a pharmacokinetic difference. Are they just processing nicotine differently? And is that contributing to the differential effects that have been seen between the sexes? And then lastly, what another fact that we found interesting is that weight gain during exposure seems to be modulated during nicotine vapor exposure in both a sex and age dependent manners, where the female adult nicotine treated animals show decreased weight gain, and the male adolescent nicotine treated animals show decreased weight gain, whereas the adult males and the adolescent females seem to be insensitive to the effects of nicotine on weight gain. And it's interesting because one of the reasons that, at least in adult users of e-cigarettes, that a sex difference that's known to exist is that females report using vaping for weight loss or weight control compared to males. And so this is interesting and consistent there, but we're still trying to figure out why it happens. So if anybody has any thoughts or ideas, we'd love to hear them. And so I know I'm a little long on time now, so I'll just end quickly now. So what we're hoping to do with this is to take these findings and to target these neural circuit mechanisms that we're beginning to establish toward treatment development for use in patients with schizophrenia, which we can first test in these animal models and then translate into patients, as well as continuing to perform these longitudinal studies to assess the impacts of sex, genetics, or adolescent drug exposure on both schizophrenia and drug-related outcomes in these animal models. I'm sorry, my kids are screaming in the back. I don't know if that's coming through or not. I apologize. And so with that, the goal here is to keep using methods like brain imaging to take findings from human studies, bring them back into animal models, and then use these animal models to develop drugs and screen them, and that hopefully we can take back into the clinical populations. And that's the same goal with using the edibles with the cannabis bought from the store, or the vaping with the cannabis that is used on the street, is to try to bridge that translational divide, or that pit or valley of death that's often referred to in terms of translational research, is to try to bridge that and overcome some of those to make that translation back into humans easier. So with that, none of this work would be possible without the wonderful graduate students that I'm so lucky to have, as well as my funding sources and the collaborators. And thank you for being such an attentive audience, and I'd love to answer any questions.
Yay! Thank you so much. I encourage people to use a little hand clapping feature, because it's always a little difficult doing these virtual, although I have to say, at least you know people's names because the names are right up on the screen. So in terms of questions, please either put your question in the chat, or you can raise your hand, and if you feel like it, you can unmute, but I can ask your question for you if you'd like to. So while people are thinking about questions, there's one already. Darn it, I had my own question. But Claire, did you want to ask your question? It's not about me, it's about you, Claire. Or do you want me to ask for you?
Hi, yeah, I was wondering why in the cannabis study, I noticed that you used pure THC, CBD plus THC, and not CBD on its own. I was kind of wondering why that is, I guess.
It's a question I've asked myself since then. And the real answer is the CBD only cannabis was not available on the Ontario Cannabis Store at the time. And I actually emailed them to ask them, why is it not available? Because it's available other places. And we can only buy from the Ontario Cannabis Store. Ontario was way behind BC in the rollout of physical brick and mortar stores, and so we didn't have them, so we only could buy from the Ontario Cannabis Store, and Ontario Cannabis Store didn't sell it. And their answer was actually interesting. This is it's like market research while also doing being a neuroscientist on the side, is they thought nobody was going to buy the CBD only, and so that's why they never sold it. But now we have gotten it, and we're repeating the study with the CBD only, and we'll report back on what the findings are. But that time, the CBD only cannabis was not available, and so that was the reason for it.
I see. Thank you.
No problem.
So in your edibles part, you had THC and CBD, or I tweeted it out. Sorry, so when I was looking down, that's what I was doing. I was just tweeting, but now I can't look at my tweet notes to see.
No, so we had pure THC. So this is synthetic THC, or dronabinol, and then we also wanted to make sure that we had. THC, cannabis is not THC alone. So the cannabis oil that we had is at 24 milligrams per mil THC. It does have CBD, but it's negligible amounts. And that's finding a cannabis oil with enough CBD is often the problem. And that's where, because of selective breeding, we've come to a place where most strains and most cannabis oils that are available on the market actually have little CBD and a lot more THC. And so finding those balanced oils is often the hard part. But Haley, who's the student that's working on that right now, is actually now trying a CBD only oil as well. And so we'll see what that brings. But the consistent finding between the THC only and the THC containing cannabis oil that's full spectrum and has all the terpenes and other cannabinoids in it still, both showing the decrease in intake was promising, in that at least it's now consistent with what you would expect in terms of a direction of effect from the GWAS studies.
But remind me again of the sex difference. So I think you saw that the females ate more of the wider spread cannabis, right? Not just the THC pure, but
not the THC pure, yeah.
But for the kind of interesting to sort of parse that apart and see, is it why that might be.
And that's what we're trying to figure out. right now. And then trying to do a head-to-head comparison between the THC and the THC containing cannabis oil. So right now is versus a vehicle. And so seeing if you put them head to head, whether those same sex differences emerge or not as well.
And the functional connectivity work, first of all, beautiful stuff. I should have said that at the beginning, but it's been since April since I've done this. So I've been a host, so now I don't feel like I've did it a good justice, but thank you. It was a really beautiful talk and really engaging, but I'm still hung up on the chicken or egg. Of course, I'm gonna ask about that. And in your functional connectivity, I think it was only males you've done, and whether you think you might see the same chicken or egg answer in males and females. And first, I'll ask you that question first, and then we'll see if there's other questions that come up.
Yeah, no, so that's you're totally right. We only had done males. We've actually once the CIHR came in, and then it took a while to get now the scanning has, I was able to hire a postdoc who's doing all the scanning at Western right now. So we've got males and females coming down the pipeline. And so hopefully we'll have that data soon. But as far as the chicken or egg, it seems like it's probably a chicken and egg question. And it's going to be able to be difficult to be able to peg it onto one factor versus another. But what we've seen so far is that an early neurodevelopmental insult seems to be changing the brain in a substantial manner and it may be making these rats more susceptible to substance use, and it appears to be similar to what's seen in the patients. But then also exposure to not just cannabis during adolescence, but exposure to cannabis in adulthood, exposure to other drugs during adolescence seems to be having an impact. And so it's, adolescent drug use changes the trajectory of the brain development, no matter what the drug is. And so if you're starting off on a certain trajectory and then you add this sort of second hit, they probably both together contribute to the final outcome.
So there's another question in the chat, but just before, and Madeline, if you want to ask it yourself, but just before we get to yours, I'm wondering, I think there's some evidence that, I know there's some evidence that in males with schizophrenia, you're more likely to have a smaller medial temporal lobe, I think, whereas you don't see the same effect in females, but it's a pretty old study. So I think it's Byron or something. I don't know if it's still thought that way. So I'm wondering, do you think, or would you suspect that you might see some sex differences in functional connectivity, right? So you see this nice correlation in the males, but I'm wondering if you're going to see the same thing in females.
And that's where we don't know, right? Yeah. So because we're capturing it right before that first, and so that's part of the reason why the sex difference is so interesting on the behavioral end, right? We're scanning them before they drink any alcohol in adulthood. Right. And that's where you saw an even bigger separation between the males and females in the alcohol drinking than you even see between the shams and the NBHL. So it's possible that those brains in the males and female NBHL rats are further apart compared to the NBHL versus
like my AirPods die, so I really can't listen. But it's my prof's thing, so I need to be there. Like she needs to physically.
Here, I'll mute. I think she unmuted herself. Anyway, go ahead.
So it's possible that those brains might be farther apart compared to even the difference between the NBHLs and the sham. And so that's what's really interesting. And so at every step, we're incorporating. Because we obviously don't have large sample sizes, especially with how expensive the MRI imaging studies are, but because we have so many sort of potential outcomes, we're now using machine learning based methods to try to see which features contribute most meaningfully to the behavioral outcome. And in this case, the behavioral outcome is the drinking. And so even if we do see massive differences between the males and females, hopefully we can identify features, whether they're the same or different, we don't know, that contribute to that behavior in both, and then we can begin to manipulate them.
Yeah, it's going to be really, really interesting. Even if you see the same behavior, which you do sometimes, you might see differences in brain, as I know you know. Madeline or Madeline, did you want to ask your question out loud, or you want me to ask for you? Madeline.
Yeah, I can ask it.
Great.
So thank you, first of all, for the talk. That was really fascinating. I was just wondering what like practical or functional effect the decreased connectivity between these brain regions has like on a patient's lived experience, symptoms, etc.
Yeah, so it's interesting. So there were two studies that papers that came out of that that study that we did in patients. The first paper was looking at the brain reward circuit connectivity, and we didn't really see much of an impact on the PANS, which is a positive and negative symptom scale that's often used in patients with schizophrenia. But another study, the subsequent study, we published it in 2018, was looking at another part of the brain. This is the default mode network. This is the network in the brain that's active at rest. When you're not particularly doing anything, this is the network that's, it's been shown to be involved in self-referential processing and other things. But that default mode network was shown to be hyperactive in patients with schizophrenia. And then once they were exposed to cannabis, or once they were exposed to the pill with the THC, we actually saw an improvement in that hyperconnectivity. And so it went back towards what was seen in the control. And that was seen to be correlating with cognitive improvements in those patients. So not in fact, the PAN scores were potentially verging on being worsened, but on the cognitive side, there were improvements with both types. And so it's the same story that, even our local field potential data is showing, that it's not just, we can't say that cannabis is bad for patients with schizophrenia or good for patients with schizophrenia. The answer is somewhere in the middle. It might help with some things. And that finding was actually consistent with some of the epidemiological findings and or clinical findings that we know that patients with schizophrenia and a co-occurring cannabis use disorder actually have better cognition compared to patients with schizophrenia without a cannabis use disorder. And so there might be something there, but it all depends on what network you're looking at, and then what the potential outcomes that you've measured and how those might correlate. And once again, that is just correlational, but it seemed to be correlated with an improvement in cognitive symptoms after that exposure to cannabis.
Thank you so much.
Thank you.
Jabron, I'm just going to ask you a very quick question, and then we'll end for the day. But I'm wondering, so you talk a lot about cognitive outcomes, which I think are coming, and the brain imaging and rewarding kind of sort of behavior locomotive, and not that that's necessarily rewarding, but what about stress resilience? Are you looking at any of those, or stress factors, or is that not your
thing? No, it is, and it's interesting. My student Brian had actually, last week we started teaching, and so it was only on Friday that I realized that this was coming up and I need to build a talk. And so I messaged the student saying, who has cool sex differences data? Send it to me. And so he just sent me something this morning. It was just too late to incorporate it. But in mice, we were looking at the effects of stress and on ACTH levels, et cetera, and the addition of cannabidiol into the mix, and see how it modulates it. And he has some really cool sex differences data, both in an open field and in the elevated plus maze, where and on fear conditioning as well, but in sort of opposite directions, where CBD seems to be less effective at mitigating stress-related changes in anxiety-like behaviors, but the ACTH levels were so different between the sexes, anyways, that it's hard to make sense of whether it's doing anything to that. But in the females, so in the control, in the vehicle-treated animals, it appears that that there are, females show less freezing compared to males, but in the CBD-treated group, that difference is not there, and so it's almost as if and this is, not just in terms of context or cue expression of the fear, but even on the conditioning day, that difference appears. And so it'll be interesting to try to figure out what the effects of stress are, but also how they're modulated by different cannabinoids. But there's some at least promising findings from this one study that tell us that yet another project to take on and yet another rabbit hole to chase down.
There's so much, right? And actually, when you look at the literature, you don't see that much on sex difference. So it's super important. Well, I have more questions to ask you, but it's 1 o'clock and I don't want to keep you any longer. I hope everybody can join me in thanking emojis or unmuting yourself and thanking Jibran for an excellent talk. Even though you couldn't answer the chicken or the egg, we kind of figured out that it was going to be both. But thank you. We really appreciate you giving your time and for, doing research on sex effects on substance use and in schizophrenia. So thank you so much.
And thank you. And if anybody has any thoughts or potential ideas or answers to some of the questions that we've because I'm sure it's the case for others as well. As when you start including both male and female animals, you're going to find differences. But it's because nobody's ever done it before, there aren't answers available in the literature. So if anybody has any thoughts or even from clinical insights or anything that you think might explain any of the findings that we have, I'd love to hear from you. So, either find me on Twitter or email me. I'd love to hear any thoughts that anybody has.
Thank you, Jabron. I have thoughts.
I would love.
I'll email you.
Yeah, that would be great.
All right. Thank you.
Maybe that'll be our opportunity to then begin collaborating.
Yay! But thank you again, Jibran, and enjoy the rest of your day, everyone.
Take care, everyone. Thank you.